Good. So here's some rules. Our friendly cameraman has run away, and I'm recording it, right? Hi, friendly cameraman. So I'm going to be the other friendly cameraman. So I'm going to be at the back of the room, chasing you around as you do your interpretive dance. Right. Couple rules. Well, we're going to limit this to 5 minutes. You can go a little bit over. Don't take the whole thing. We have a lot of teams to cover. I want to show you what we have in our building. And then we want to come back and finish up with a survey. And then just a recap of the whole week. In terms of presentation, there's a little doohickey thingamajig here. Number 2 input is the computer. So if you want to load up your presentation off of Google, that's number 1 input is the Elmo. The Elmo has a zoom setting. So this dial up here. And then you can press the button on this side to make it focus, all right? So whenever you're sharing something, put it flat on the surface, zoom in, and then focus in on it. Okay? Okay? Whoever is speaking, whoever comes up, first write down your team name here. Otherwise, we don't know whom to vote for. It's up to you. You can have just one person come up, or you can have the whole team come up. It's probably a good idea if you're trying to make a bid for the team.
Okay, listen, please.
If you're trying to make a bid for the team prize, you might want to have everyone up, right? Okay. So I leave it up to you, but please put this on whoever's speaking, or share it between whoever's speaking, and please put your name here as soon as you come up here. Okay? Questions? Does everyone have a voting sheet, a voting ballot?
Oh, wait, where's that?
Okay. I guess you guys want to go first?
Who's talking first? I think it's Miriam.
So, do I just... Okay.
Yeah, there's nothing to turn on.
Okay, and here we go. We have 1, 2, 3, 4, 5, 6, 7, 8 teams.
There's a pencil.
Milady. Okay.
Read, man.
So we're Team Milady, and our design objective was to create a device that can tell if milk has spoiled by determining the pH, if the pH has reached the threshold that classifies it as spoiled. Our design criteria was that we wanted to make a relatively simple device that would be small, cheap, portable, effective, easy to use, and easy to make, and also a single-use product. So our first prototype, that's not our first prototype. Okay, yeah.
This one.
So you would add a few droplets of the milk at the to the circle. Yeah, to that circle. And then ideally the milk would travel to the long oval section to be filtered, and then divide into the 2 other circles where where there would be 2 separate readings of the pH. Then the extra milk would flow to the last circle and then empty out into a separate glass. So when we actually tried this, the milk would not enter the long oval section and instead leak into the layers of the device. So when we moved our device around a bit, the milk actually entered the oval section and then indirectly and slowly reached the pH, and we had a reading. But then we also decided to make a second version of our original design, and this time we accidentally overglued so the milk wouldn't enter at all.
Okay, so from this we learned that it's really important to seal the edges of our device so the milk doesn't flow through the spaces between the plastic. But we also learned it's important not to overglue because it'll block the channels that the milk has to travel through. We also learned that we didn't really need the long oval section, as it just complicated the device and was unnecessary in completing the task. We also decided to put filter paper and pH paper all along the device, except for where the milk is first put, like so. Yeah.
Yeah, see how it's yellow all the way through? That's filter paper to wick it.
Yeah, so that we could whip the milk. We made minimal changes to our original design for our final prototype. We removed the long oval section, widened our channels, and put pH paper and filter paper throughout the device to wick the milk. We also made the device 6 layers of plastic thick and made sure that there was just the right amount of superglue used. Finally, we put a barrier layer of plastic so that we could stop glue from interfering with the device's function. When we tested this, the milk pretty much flowed its path to the filter paper and then to both pH papers.
Okay, so for all the reasons that Lydia just mentioned, that is why we deserve the most improved design, because we started with something that didn't really work, and then we got up with something, and it did work in the end, yay. So, also, it could be most manufacturable because it isn't very difficult to create, because it's pretty simple and pretty symmetric. We deserve best teamwork and best name because our name is Malati, which incorporates all of our names, and it just fosters the team atmosphere, and also it sounds really nice, like Malady. It's nice. Okay, and then I could tell you why we deserve best presentation and best overall product, or you could watch our infomercial. So.
You all did an infomercial, correct?
I know, I'm gonna get it. It's called like No, it's not. It's called Malady. Get it right. See if I can. Oh, here it is. Okay, is the sound on?
You made it.
Is the sound on? I can't tell. I think it's on.
There's nothing more refreshing than a glass of cold milk on a hot summer's day. Oh yeah! But when you can't tell if your milk is expired or not, it's not so refreshing. Who can tell if it's safe to drink? Not even Mr. Whiskerbottom knows. Don't you wish there was a way to tell if your milk is spoiled? Yeah! Well, with the new Malady, there is. It's super easy to use and super effective. Just pour some milk into Malady and see if the paper turns green. That simple. Wow! Malady has patented milk sensing technology to move the milk to the magic paper, which turns green if the milk is okay to drink. And now Malady can be yours for only $9.95. But wait, there's more. If you call in the next 90 seconds, we'll double the offer. That's we'll send you 2 Maladies for the price of 1.
Just wait, we're crossing in hands.
That's a $30 value. Yours for just $9.95.
Product label to individual steps, item processing and handling required. That is been include John's flooding technology, dehydration, food poisoning, swamp, they have you have to use Malady if you all were planning on becoming accredited board. Malady is not used by smokers, drugs, or with any school load and food. Malady is not responsible for any washes or neck. To order, call 1-800-921-7753. Once again, that number is 1-800-921-7753. Malady is trademark of My Vision Appropriate.
If your milk seems spoiled but hasn't started to smell, pour it on Malady and you can tell.
Malady.
All right.
Yay!
All right, who's next?
Next team up. Volunteers? Who'd like to volunteer? This is where you raise your hand. This is where I say who would like to volunteer, and everyone does this. Look at them. See how he's doing that? Okay, so who wants to do it? Thank you. Well, should we do a triangle? I think we will. So we start over here.
I'm doing so well looking down too.
Please hold up the microphone. Write your name.
Bye, you tell me first. Yeah, you got it, right? Yes, you can get the mic. Okay, so okay.
Okay, so we
And
are.
everybody, please be mindful of the fact that the microphone will pick up sounds that you make, sometimes preferentially, it appears, to those that come from the people in the front. So please try to be quiet. You did great in the first part.
Okay, so we're team no, and so basically we would like to be. I'm nominating our team for best implementation strategy and most synergistic, harmonious team, as well as most creative name. Okay, so the objective of this project was to create a biosensor that would determine the spoilage of milk, and the design criteria for this project was to create a biosensor that was disposable, easy to use, easy to manufacture, and was also cost effective.
So in our first prototype, which is the one at the top, we have a full thing. So it starts, you put it in the well, and it flows to the filter paper and then to the pH paper. And up in the corner, we have the pH papers of spoiled milk and good milk. So it's easier to compare which one is which, so the consumer can tell which is which. So for our first one, we had a couple of problems in that we didn't have enough glue, so the milk pretty much just bypassed the filter paper, although it still eventually made its way back to the pH paper, so we still got a good reading. So for the second trial, we added more glue and tried to use less filter paper so that we could have less constricted channels. And this was more successful, but we still got a lot of the milk that didn't make it where we wanted.
Okay, so in our third trial, to solve the problem that Robert mentioned, we allowed Lyle to basically lather the layers with glue, and we didn't worry about filling the channels up. And then once we glued it, then we re-cut out the channels, which widened them and made sure that they were not blocked by the glue, and also kept the pH paper from getting glue on it. And that worked much better, and the glue stayed on its course much better the last trial. And in the first trial, as Robert mentioned, there was a lot more filter paper. So the second trial, we had the same thickness, but we just had a lot less surface area. And that worked well, but it still was too slow. So we tried actually having a very thin piece of filter paper, but it was much longer, and that worked well and filtered it much better. And so yeah, those are our 3 designs.
Thank you very much. Another volunteer, or is he with a triangle pattern? There we go. Oh, Joshua, you want to make sure you put your team names.
All right.
Okay.
What are we doing? Just our names.
You can choose, yeah. Oh, goodness. Oh, wait.
Do you want to press anything? Okay. So if you want to know, okay, so the goal of this biosensor was to detect milk spoilage by recognizing the decrease in pH level indicated by the change in color in the pH paper. Our design criteria were to be aesthetically pleasing. It's easy to manufacture or mass produce. It's easy for the consumer to use, and it's cheap. So our first design was originally like this, and as you can see, we cut off our top one because we could quickly realize that the serpentines were a pain in the butt to cut out, so we didn't do this part. And the reason we had these 2 channels was because we wanted to see the difference that the amount of filtration would do to the filter to the pH paper. Is that? Yeah. So we couldn't even get the milk to go into our 2 holes right here, if you can see that, because we had used too much glue and it blocked off this part, and the filter paper soaked in some of the glue because we didn't wait for it to dry before putting in the filter paper. And even if we had been able to, I think that the passageway was too complex for the milk to flow through efficiently, so we wouldn't have gotten down to here. So, do you want to talk about our?
Yeah, so it was pretty evident from our first trial that it was way too complicated with 3 or 2 serpentine blocks. And so our main goal after the first trial was just to create a design that would have one direct path, making it extremely easy for the milk to get to the pH paper. And so, but at the same time, we still wanted the design to be aesthetically pleasing. So we came up with a design that kind of looks like something you could use for a company's logo. So in our case, we just use our initials, but we were thinking, oh, we could have like the company's initials or company abbreviation as part of the design. So basically, they're letters that have one direct path, and it was convenient for us because the wedge of the Q or the pocket kind of created a place for the pH paper. So we think that this works well as a most improved prototype, most improved design, just because we went from extremely complicated and unnecessarily complicated to simple and functional.
Sleek and still aesthetically
Yeah.
pleasing.
Yeah, and most manufacturable because just us gluing and cutting, it was a lot easier to, I guess, produce the second one. Yeah. That's it.
Thank you. 4th group.
Bouncing back to that way. Or I guess Donna and Ivy.
I can tell from their eyes that all 3 of them are ready.
And
You don't have your.
And here we go.
Okay, so our name stands for microfluidic indicator that detects the level of change in pH.
Okay, so our design objective with this project was to build a sensor that can detect the spoilage of milk, and we use the pH level in the milk to detect the spoilage. We wanted to make sure that our sensor was disposable, easy to use, inexpensive, portable, and easy to manufacture.
So our first prototype was successful in that the milk was able to reach the pH paper. Right here, our first one, it basically differs from this one as there was a serpentine path right here. And basically the milk got to the pH, but it just like felt like it went through the entire prototype, like just seeped in. And it got that. There were some complications because it was really hard to cut it out. It took a long time, and there was milk seepage into our device. We learned that the simpler the designs are better and that they're more efficient and take less time to make. We also learned that we should have sealed the layers better so that the milk would just stay in the canals we made.
So in our final design, we made improvements to fit with our lessons learned. We cleaned out the canals and made it thicker layer-wise. We made it 6 layers. So we glued in more appropriate places, and the filter paper also was used more sparingly, so it wouldn't clog up the biosensor.
And we would like to nominate ourselves for most creative name, and most synergistic or harmonious team because we worked really well together, and it spells out milk, kind of. You know, so yeah, thank you.
Yeah. Okay, 4 teams. Team number 5, up we go.
I drew it. Do you want to write the name? Yeah, sure. Okay. I don't like it.
Just on the spot.
Yeah. Oh, problem with it, Robert? No. Oh, problem with it? Just slowly going that like, we got it. Okay. Okay, so we are Donna and Eileen, and this is our first prototype, and that's our second prototype. And so the obvious problems with the first prototype is that we originally had a different design, but due to miscommunications, this was all we got. So you would basically, the idea was that we would have a lot more complicated stuff going past this point, like we had kind of like a square sort of thing, but then in the end we just got these 3, and you would put the milk here and it would travel through, and then it kind of just fell out, and so we had to clean that up. And so with the serpentine, we discovered that it was annoying and pointless, so we got rid of it in the second design. But also the fact that the filter paper, as we glued it on, Katie's group used way too much glue, so then the filter paper got soaked up in the serpentine, so the milk never actually made it through. And we put the filter paper here, but that didn't really gain us any results.
Yeah, and we kind of ruined the pH paper by doing that too, which didn't really matter because it didn't go through the whole way, but yeah, so we learned better. And then for our second one, we decided to do something a lot simpler, so we just used the chambers instead of the serpentine, so that was a lot easier to cut out. And then we decided to have 2 chambers of pH paper in them because then we figured you could test it twice, and then if one didn't work, then you could tell because the other one would be a different color. And we located our filter paper up here instead of having it throughout the whole thing. So that way we wouldn't make the same mistake, and even if we overglued somewhere, then it wouldn't soak into the filter paper and ruin it.
And another thing that we did about the filter paper was different was that we cut out a big rectangle, and so we super glued the sides instead of the part that the milk would actually travel through. So then so even if we used too much super glue on the sides, the milk would actually not be affected. And we also did the same thing for the pH paper at the 2 ends. And it worked. So you vote for us for most improved design.
This is group number 6. There are 2 following this one. Oh, and feel free to erase what's behind you because as you talk, we'll be looking at it.
Okay, hello. We are the Dream Team, and our objective was to design and build a biosensor that detects the spoilage in milk. Our design criteria included having the biosensor be small and portable, efficient, easy to use or simple, cost-effective, and successful.
Okay, so for our first prototype, it already was pretty simple. It kind of looks very similar to our final prototype. The only difference really was that we had just filter paper on one side. So we would just have it on this side right here. And so basically at first, before this, we tried to implement a serpentine, but right from the start we knew it would just be too complicated, and so we didn't. And then, but for our first prototype we did make some mistakes, like for once at first we glued down the pH paper. Lesson learned, don't do that. And so that kind of blocked off the pH paper from reacting with the milk. And we used a lot of glue on our first prototype, so we blocked the channels. And so, yeah.
Okay, so when making our final design, we didn't really like switch up the way it looked, but we just worked on fixing up the pH and filter papers as well as our usage of the glue. So we had to make sure that our channels were clean because a lot of the time the milk couldn't get through. And along that line, we also had to like make the channels like wider, so because as the days progressed, the milk got more spoiled and it couldn't get through the channels that were that thin. We also had to make sure like it was glued all around, but not too much, so it wouldn't also leak. And we also, the pH papers, we used 2, so we could have like a more accurate reading, and that did like prove useful like during our trials.
Okay, so because of this, we would like to nominate ourselves for the like manufacturer of the simple one, because I think ours just goes down and out. So it's pretty simple. And then we also like to nominate ourselves for the teamwork one, because we all had like our separate jobs and it really worked well because we got in the end 3 prototypes done. So yeah.
Thank you very much.
I'm not going to the cheat
Good job pulling, though.
We don't need anything in this. Just put this one. Leave it. Okay. Alright, so the design objective obviously was to create a biosensor that was able to detect spoiled milk by checking the pH running through a set of filters that you can see there.
Okay, first of all, name your project.
Oh, we are the pampered cows.
Because we produce spoiled milk. Ooh. Okay, so our design criteria was basically what most of you guys already said. It had to be portable, easily manufactured, and distributed, and it would also work for a small sample of milk. And so basically the theme of our project was we kind of had grand ideas, and then we slowly, after prototype and prototype, we just learned to cut down. So as Nick's still drawing, we have 6 total prototypes, and Nick's on number 3, I believe. So we'll just start at 1 and see if we can catch up. So our first prototype was to use a serpentine. I don't know why we used it. I think we thought it just looked cool. And our first idea was that the milk would go through the serpentine, and since the milk would go through different concentrations, and so it's eventually streamlined, that did not work, and it just got stuck in the serpentine. It didn't go anywhere. And so our next idea was basically just took our prototype, cut out the serpentine, and put a big square, and that worked. That actually worked. Oh, well, it is a square. Yeah, so it was a square. We put a filter paper in there, and then a pH paper in the next square, and that worked, but we thought that this kind of looks boring. And another thing that we saw was that the milk would get stuck in the channel, and so we thought, why not make it easier for the milk to pass through? So how about you go?
So after that, we decided we need to widen all the channels, as you can't really tell in the designs, but
got the kind of tapered.
At one point, we figured out, I think it's the 4th prototype.
Yeah, go.
The 4th prototype was, the milk wasn't moving through the device very well, so we figured we could do 8 layers of plastic, with the first 6 layers being solid for the well, and then the top 2 open. And then after that, we do the first 4 layers, and then 2 open, 1 closed, as a gradient all the way down to the pH, hoping that the gravity will help the milk move down towards the pH paper, along with filter paper. But our problem was it wasn't moving fast enough still, so we had to tilt it with one of the razor blade caps.
So it still, again, it worked, but not very well. And so we thought, we're not even really using this. Why not just put in the pH paper here and the filter paper here and just cut this part out? So that's exactly what we did. And if you guys remember on the first sheet we got, that was one of the designs, that just a well in the middle piece and another well. And when we first saw that, we laughed at it and like, oh, no one's gonna use that. And then we used it. So, and then we went one step further and got rid of the well. And so we have this like nub thing with our filter paper in there and our pH paper. And that worked too. So I guess we learned that simplicity is elegance. And so, we're gonna nominate ourselves for a couple awards here. We're gonna go for the most creative design process and concept. Most improved design, I think, just by the sheer number of prototypes we went through. This was an example of trial and error. And also teamwork. You see we have 4 people here, and you think, oh, multiple times people came up to us saying that you guys should be done with 4 prototypes now, and we're still halfway done with our first one. Well, one more person is another person to organize, and some special people are more fun to organize than others. So
we love each other.
We love each other, and we all. This was planned, but again, so I think we came up with an okay design, and I think the greatest thing is that we learned a lot of what a biosensor should maybe be. So, yeah. And most creative name, I mean, it's a pun, so not really. Okay,
Yay.
yeah. Pampered children are spoiled.
Somebody told me. I know, we all heard it. Who called you?
Wait, I'm going to the prototypes. Well, you're speaking first. I know how to do it.
Alright, so we're JDS Industries, and our design objective was to create a biosensor that detects the quality of milk by measuring pH. We wanted to make sure our biosensor was portable, easily manufacturable, disposable, aesthetically pleasing, and easy to use.
Okay. So our first prototype was we had a well, and then we had one of the, I guess, the tank blocks, and then we had our serpentine block with filter paper in it, and then the milk would go through that, end up in another tank block, and then end up in this well, which has pH paper in it. And obviously, the first one we ran into was the serpentine block. It didn't really work, kind of like how it didn't work for all of us. So we decided to get rid of that, and then so for our second prototype model, we just put 3 tank blocks in a row because it looked aesthetically pleasing and it was symmetrical, so that was nice. So then we basically followed the same design and put filter paper in the middle, and then we put pH paper at the end, and that worked better, I guess, but the biggest problem was that the filter paper was absorbing all the milk, so it never actually reached the pH paper, and also our gluing was pretty bad, so the milk just kind of seeped out. And yeah.
Alright, so the lessons we learned is like, first, we don't want to incorporate the serpentine. We want to make it as simple as possible. Then also, The milk would had a lot of trouble in the first design, especially getting past the filter paper because we used too much and it was clogging up the path. So we concluded that it was best to avoid use of the design, that design, and instead we used this, as he said. So we also cut the filter paper to fit the design, and as he also said, we used a 3-layer design in all our prototypes, but in hindsight, we believe that if we had more layers, it would allow more space for the milk to go through easily. Another thing that obviously would have helped is if we'd done a better job with our glue jobs and if they'd been like perfect, but you know. Yeah.
That's everybody.
Yes.
You got that in some form?
I have the video right here. So maybe I can project it.
That'd be great. Okay, thank you, everybody. Take a look through those ballots. You see the names on there. While you're working on that, we're going to get something put up. We're going to show you something that one of the freshman teams developed that we think you might find to be interesting.
Yeah.
Sam Blow. Sam Glow.
Oh, I guess it's Sam Glow. Sam Glow. No.
No, that's Nobody's a pregnancy test.
Okay, here. So I'll turn the lights up.
Yeah.
Here's a device that I think you'll find it here. I'll turn off the lights back here too.
So, just by way of background, I think one of the lessons that we learned as educators is that creativity doesn't depend upon age. And the freshmen were equally creative. I mean, they learned exactly the same lessons that you guys came up with. So it must mean that you guys don't really learn any engineering in school, right? But, or I guess I should say that engineering is rather accessible. So, why did this? Hold on, let me make this really bright. There you go.
That's a little very quickly, can't you?
Yeah, I guess you might want to turn that on. Okay. I think it tricked the sensors. We're talking multiple sensors here. So let me just go back here. Whoops. Oh, that's my newborn. And that where is that video? There it is.
Can I try one after this?
Okay. So they made 8 prototypes in half an hour and tested 8 of them, and they got 8 out of 8.
So whose idea was this? Yeah.
All right, so we hired that guy to work in our lab. I guess the point here, one of the key things they recognized that milk gets everywhere, so they eliminated anywhere that they didn't want it to go. So essentially, they just glued 3 strips on to make those walls, and the 1 strip in the middle, and that was it. It's just a single strip glued on, so there was no gluing layers, very, very minimal glue application, and very shallow but wide tracks. So they actually exploited the fact that the milk gets everywhere by creating those barriers around this thing. So it was a particularly clever design. I think it was something pretty inspired, we thought. But it's clear that you guys reached the same conclusions. You recognize the importance of repeatability, redundance, the ability to actually make these things easily, so the design for manufacturability, by having multiple sensors in there you thought about improving reliability of your sensor, and all these things are really good, right? So while keeping those cost considerations in mind. So I think you'll see that you have independently discovered the KISS principle. How many of you have heard of that? It's called the keep it simple, stupid.
I told you.
Yeah, so it's called the KISS principle, and that's what you discovered. And the next thing, you take your 5 minutes to write down your thoughts on your sheets of paper. We will tally those numbers, and we will share with everyone who won what award. And then after that, I'm going to take you around the building so you can look at some of the stuff that we work on, which is very similar.
When you're done with your ballot, let me know, and I will take.
Right now, but the idea is that, and you can see pieces and parts hacked together from the hardware store. So here we have just drawer rails that you might find in a nice smooth drawer, and this armature is going to move back and forth. It'll be pushed with a motor, a linear motor that serves as a muscle moving back and forth. And then we have this roller over here that will move up and down, that's connected to this armature. It will move up and down and crush the cysts. And we have plumbing system of water that will hose down the top and fire off jets from the bottom and rinse the mesh from the other side. And these are gutters to carry away the waste. We have pieces of plastic that we bend using the bender that will sit here, and this thing will rock back and forth like a seesaw. So whenever we're just getting rid of junk, tip it back into the gutter position so everything flows into the gutter. When we want to collect the sample, it flips forward and the sample rolls into our sample cups here, so we don't collect too much junk in the sample cups. And the way this, I guess, upstream of this, we have this thing, which is probably recognized from the hardware store again. This is the inside of a toilet flush. This is the thing that keeps the clapper on top of it, and there's a clapper down there somewhere. It's hanging off of that. Yeah, so this is again 4-inch pipes straight from the hardware store. This is where we make our dirt milkshake. So the operator comes and puts a plug of soil. We have a paint mixer back there and a motor that will drive, basically there'll be a jet of water to mix up the dirt. The cysts will float to the top. We add more water, the cysts overflow onto this mesh where they get crushed and processed. So right now, humans do this job, grad students. We want to make a better world for you, and when you go to grad school, literally people get their degrees just crushing cysts for their entire thing. So it's a pretty painful way to do grad school, right? And so we want to eliminate that. And while we do that, we want to make the process of testing 100 times cheaper. Because you take away the person and you make this stuff out of such very simple parts, you can afford to do things like that. And then you really can expand the kinds of things that you can do. Right now, a farmer takes 12 plugs of soil from 12 acres, pulls them all together, and gets one reading because it costs $50. It's really expensive. What we want to do is give them 100 readings, so it's the same amount of money. Right? And that will really improve how they deal with the soil-based network problem. So those are the kinds of problems we solve. Points I want to mention, one was that we build our own tools. We build prototypes such that they can be prototyped quickly. So like if you notice, you never see a real machine that looks like this. But when you have so many holes, you have many options to adjust your dimensions and get things in place and so on and track. And you can take these apart very easily and put them back. So really, we're playing with Lego blocks. They just look a little bit different. We have here some biosensors that we've been working with. So these are actually real biosensors with real microfluidic channels on them. This is the disposable part of it. The principle behind this biosensor, it's called the surface plasma on resonance, is the optical version of skipping rocks across the surface of a pond. Right? So it's if you bounce light off of a surface, part of it gets, or most of it I should say, gets reflected, but a little bit of it sets up a ripple along the surface. Right? And at some angle, that ripple is the highest you can get. So most of the energy or greatest amount of energy goes down the surface, and the smallest amount comes off as a reflection. right? So inside this device, you can see it's just very, very easily manufactured, very cheap. Inside this device, you have a plastic prism, an LED, and a sensor that resembles your digital camera sensors, except those sensors are 2D. This one's just a one-line sensor. So you can actually see where the reflection comes. You can measure the intensity of light along that line, and wherever the light is the smallest, you can calculate that as your angle. And here's the microfluidic chip that brings the sample. So what you do is this is a biosensor, so you put a recognition layer on that gold surface, something that will bind to the molecule that you're interested in. And the refractive index, the change in the refractive index is the transduction. Right? This is the microfluidic channel that brings that fluid. So you basically stick your sensor in here. You have two tubes running into this port. And notice it's a plastic block with a Teflon or actually a silicone gasket on it, and the gasket has one little channel running in it. So when you push that sensor in here, it seals up against the gasket and you have a single channel. So as your fluid flows past that single channel, your gold surface on the sensor grabs the analyte and then transduces your signal. So you can see that very, very simple designs, very simple motifs are everywhere, and we really make use of this. So I'm really glad that you recognize that simplicity is key. It allows for fewer failures, and you can get a more robust, more functional device that way. Right? So the rest of the stuff we do here is some electronic stuff, some testing equipment, and then I'll show you my other lab where we do most of our molecular biology, but we kind of grew out of this lab very quickly. Questions? Let's go. So when students come into our department for the first year, in the freshman year of college, one of the courses that they take is maybe 100, and that's a design project. So very much the same kind of thing, except that groups of 3 or 4 are working on different design projects. And some of them are science-based, which means that they do an experiment and find a result, or they are development-based, where they'll make a prototype and build something. So here on the wall here we have the most recent team's efforts translated into posters. And you'll see the same kind of theme running through. We sort of insist on one-page, visually attractive descriptions of your project. We don't care about the large volumes of stuff that you would write. If you can communicate in very small amount of space your objectives, your methods and conclusions, and your data, then that's a really important skill to have. Very few people have time or interest to read large amounts of things, right? We're all a Twitter culture these days. So this is an example of some of the, I guess that's my project that I helped with, the effect of pH on bacterial resistance by the sourdough boys. This is an example. How many of you have had sourdough bread? How many of you have made sourdough bread? Or friendship bread, sometimes it's called. Yeah, so these are cultures that have been passed down for generations. Bread cultures, bread inocular. And the cool thing about these things is that this is 10,000-year-old biotechnology, and it's robust as anything that we can ever do. You cannot spoil sourdough, right? It's just a self-replicating, self-sustaining culture, and it continues to give the same service, that is, produce good bread year after year after year. I have a sourdough in my fridge from 6 months now. So one of the things we're trying to do is understand how is it that these communities of bacteria are so resilient to invasion. How come something doesn't just jump in and set up shop and spoil your sourdough culture? Right? It's kind of the same reason why once you turn milk into yogurt, it doesn't spoil as easily as fresh milk does. So it's sort of the same reasons. So here my students have been looking at what makes sourdough so resilient. So they've been spiking it with other bacteria and seeing that those bacteria actually don't survive too long. And a bunch of it is because of the pH. You reduce these lactobacilli reduce the pH down so quickly that potentially harmful bacteria don't have an easy way of making a hole there, having a niche there. Right? So let's go downstairs. Now, lead away. No, I found it. Okay, we have an important announcement. Students take 3 courses and a written testimony that they have taken those 3 courses before they can enter. Okay, my students. So this is the only biosafety level 2 lab in this building. One of the many biosafety level 2 labs around campus. What this means, so the normal stuff that you can do with E. coli and all that is biosafety level 1, which means that you know you're working with biological organisms, but they're not likely to cause you any trouble. Biosafety level 2 means there are possibly some organisms that might make you sick. Okay? So one of the things we're working with here is human waste from the Champaign Sanitary District. Cool?
What about what's level 3?
Don't lick the floor and you'll be okay. Right. Biosafety level 3 is for known harmful pathogens. Right? So if you're working with Ebola, no, Ebola is biosafety 4. But if you're working with HIV or hepatitis B or something like that, it's a BSS lab. Right? So the reason I mention this is because there are some protocols that we have to know about. Everyone wears gloves and coats inside, face masks, works inside sterile situations and so on. We're not going to do that with you guys right now. Just don't touch anything, right? I'll show you where the hazard is, and as long as you can stay away, as long as you can keep your hands in the pocket, that's right. You're going to be fine, okay?
It smells like push. I'm not that desperate yet.
Go ahead. All right, that's good. Alright, so I want to introduce you to my friend the Jugaad. So, those of you of Indian origin might have heard the word Jugaad. Yes? Yeah. So, Jugaad means a hack job. It's a solution from obtained from the materials at hand. You're looking at it right now. So, when I was starting out, I didn't have that much money. I really needed a photobio reactor, a chamber in which I can put light in and I can get my plants come to grow. Right? The stuff that I could buy was essentially a box with one of these lights inside, and it was they were going to charge me, and therefore you, dear taxpayers, $6,000 for it. And so my grad student and I went to the local steel store, bought some sheet aluminum, and hacked together this stuff in an afternoon's work. Okay, so this is my photobioreactor. I told you about our nematodes. The nematodes infect soybean plants. We use this chamber to grow our soybean plants. We use this chamber to grow anything that requires light to grow. So our plants come, we use this for. Our nematodes, we use this for. Very simple, just lamps inside and computer fans to draw out any hot air. So if the temperature goes above a certain level, the fans just come on and pull out some hot air. Okay? So we call that the spirit of our lab because we really believe in this kind of stuff. Produce your own solutions when you don't have anything to go with or you don't have the money to implement it. Right? So hopefully that's a lesson that you guys learned as well. Now make sure you're standing closest to the hook. Right here. So this is our biosafety level 2 restricted materials. You can see they're all in serum bottles, they're all sealed. You're not going to come in contact with them unless you kind of have a bar fight here and break these bottles. But never mind. So delete that. So the thing we're studying here is we're trying to study the impact of nanoparticles on the environment. A lot of people are using more and more nanotechnology in their toothpaste, in their deodorants, in their shampoos, for their wall cleaners in the bathroom. the shower on, you step in it, it gets washed off, and goes in the drain. In the sewage processing facility, microbes, again, are providing us with an incredible service. They digest all that junk for us so that we can discharge clean water out into the environment. But if we now start putting all these other things, these nanoparticles, in the environment, into the sewage system, what do we know what's happening to those microbes? Can they survive it? If they don't, that represents a failure of that sewage system process, and we will be discharging literally poop into the waters. It's not a good idea. So that's what we're doing. We're actually taking these samples in, subjecting them to all sorts of nasty things, and looking at how their colony composition changes, how their function changes, and trying to analyze how can we measure this, how can we build a system that can flag some problems very quickly, and so on. So much of the equipment you see here is related to molecular biology. As you might imagine, we're using a lot of things that grow. So we have 6 incubators. So there's 1, 2, 3, 4, 5, 6 incubators in which we can grow bacterial cultures, mammalian cultures, and plants. Right? Some of the other pieces of equipment have to do with extracting DNA, amplifying it using PCR, and we have a couple of safety hoods. This is one safety hood. There's another one on the other side of this wall where we can do our bacterial work. So this is really a key piece of equipment in a biosafety level lab. So you can work behind the glass in your gloves. There's an air curtain that's making all this noise, and you can keep the environment clean. So there's filters in there, this case surface every year, and so on. Right? So this is our clean biosafety hood in the sense that we do our careful work here, and the waste sample stuff we do in the other biosafety hood. Clean doesn't mean the material can handle it. Clean means we clean it down so that we don't cross-contaminate our different experiments. We're running many experiments, many students are running experiments here, and we try to minimize the risk of one student's work affecting another student. Okay? So if you want, you can walk by this way and then out the door, and I'll show you some of the other things here. To solve problems that other people haven't been able to solve yet. Alright? So if you come here and work for us, we'll first probably put you as the dishwasher. Then that guy doesn't work as the dishwasher. Alright? And then you can work your way up. Alright? Yeah. Exactly how it works. Alright, let's see what we can open. Look at my key. Just So this is our hydrology lab. Some of the things that you will do with filter papers and microfluidics. This is the grand, large scale version of it. Now I'm standing in the biggest shower that we have on campus. You can see the shower curtain that goes all around. The shower comes up from there. So this is actually a rainfall simulator. And you can simulate how rainwater seeps through different kinds of soils, what angles you need to have your soils at, how much erosion you can create because of this. And you'll see that the mechanics of this problem is much the same as pigment flowing through liquid paper. Very similar ideas, capillary action, diffusion, flow, all those kind of things. Back there we have a large fluid plume in which we can simulate waves. So we can actually simulate the action of waves on that one. When people get solved problems from, what happens to war gained areas. So there are one of our projects that my colleague has done is for the army, where they roll tanks on these large areas and they compact this area so that stuff doesn't grow there and the soil gets really compacted. How do you restore such a piece of property? Eventually this gets really, really ugly looking over time. So those are the kinds of problems they try to research. They're related to mechanically the same concepts as within sort of the microfluidics exercise. So one principle in engineering is this underlying math that doesn't care which particular system you're working with. It's just as applicable to tiny little pieces of filter paper as it is to biofilters and rainfall. Let's go and look at some drones.
Great idea. You gotta go. I just found this room, when the door opened in just a second.
Or you want to say? I don't know. It's my one. But again, here is good stuff. Most of those businesses like that. Let's go. Actually, wait, let's go across. There's very private space back there. So let me see space. One of the projects that students work on, maybe I'll explain it here and then you can take turns doing it. So one of the projects that students work on is the pullers, the tractor pullers. So it's kind of like the competition we had in this 1 week, except it's a year-long competition where students are given 5 engines and 4 tires. And they have to build a tractor with it. That's all they're given. And they have to compete for who can pull the most weight, who can run the fastest, and who has the best manufacturable design, and who has the most reliable design. Stuff happens, and they actually compete, at the event. Things break, pieces fly apart. So all kinds of crazy stuff happens. So I'll show you some of our tractors that the students are building on right now. Space is tight, so take turns coming in. They're given the engines and the tires, and they build everything as low weight as possible. maximum power to weight ratio. The design is very much like a prototype. Very little refinement, a little bit of instrumentation to measure how they're doing, but essentially it's a fully working automobile. And they compete every single year. As lightweight as possible, as easily manufacturable as possible, as quickly as possible. They have all the tools to do that here. Clearly, they're driven on soda. Right, let's go back. If everyone see, I guess let someone else come in and take a look at it. Right, let's go this way. It's a bigger group, so walk this way.
Are they gonna be really?
Oh, cool. Little extension big.
Everyone is. Alright, so this lab is the agricultural remote sensing lab. They've been doing drones before drones were cool. So essentially this is one of their oldest models. There is a new model which is a quadcopter version on the other side. So one of the applications for these things is to fly over a field and look for stresses in the crops, see whether the plants are getting adequate fertilizer, adequate water, and as these fly over, they can get the GPS signals, they can capture a picture. So this is actually the camera gimbal. So as this helicopter flies over, it can pan the camera around, take pictures, and make sense of that data. So this is, as you might have seen in media, drones are becoming incredibly popular for all kinds of different uses, not just military applications. And one of the areas that we will see a lot of is in agriculture. We have robots flying in our skies, looking at what needs attention, what's doing well, what's not doing well. Right? Okay. Let's head back upstairs.
I will go through. I don't have an envelope. It's kind of like an envelope. So I will announce who the recipients were. Oh, thank you very much. And so once we do this, there is a survey that we would like you to take. And after the survey, I think we're done.
Yes, we are. And before we announce the results, I would like to say that I had a lot of fun making this stuff. So we've actually, you might think it seems a little silly, cutting up exact pieces of paper and gluing these together. But in fact, one of the key steps in our image acquisition robot that we're building over there is the idea of taking a sample, putting it on something, and then imaging that something very quickly and in a disposable manner. Guess what we're using for our prototype? Plastic. Transparency film. And actually, I'll see if I can hold this up, but our prototype doesn't look very different than what you guys were coming up with. I suppose it's a little bit simpler. Essentially, it's a, do you know what a hemocytometer is? It's a device, it's a cell counter. It's basically a little device, it's a glass slide that has two ridges on it, and you place the cover slip on top and you squirt a little bit of fluid inside. The capillary action holds that fluid in a square, and you put it on the microscope and you count how many things there are in that square. So we essentially built that out of this same kind of material. That's blue ink in there, that's all. But you can, because of the laser printability of that sheet, I can put a barcode next to it, which a computer can scan and read very quickly. So that barcode says, hey, this is Farmer John's acre number 32 60-second sample, right? And my computer can read how many cells there are in that chamber and analyze it, and I can destroy this very easily, right? And it's made out of the exact same material that you use. So, coming to a Research One university near you, the state-of-the-art microfluidics technology, right? And you guys gave me some ideas about how I might improve my design. So, I think all of you are winners. You all did really well. Yeah, everybody says that, right? Okay, we'll get to the real winners.
Okay, the winners are, and we are all winners, most creative design, Quinn and Josh. This is where I should give you something, but I don't have anything.
I think we should say something about it.
Okay.
It's. No, we should say something about it.
We should.
I think you thought ahead about marketability of your product, and you came up with an idea that somebody else could make their own, and somebody else could have ownership of that design. Right? And that's thinking at a much higher level than actually just solving your immediate problem. So kudos to you guys.
Excellent.
Though I might say your design came out of vanity, but that's okay. So did everybody else's.
Okay. Best implementation strategy. I need to work on pronunciation on this. No! Or is it new? No. No. It's like the moo with an N. Okay. Do you want to add?
I'm trying to think of what it was.
Okay.
But obviously, the crowd part, your implementation was the best, so kudos.
Most improved design, pampered cows.
Yay.
Okay, that's most okay, so most gratuitously complex design went back to design number 1, Quinn and Josh.
Also, kudos to make me make parts that weren't on the original list.
Okay, most synergistic, harmonious team, we have a tie, dream team, and milk.
Yay.
Best documentation or presentation, and this group had 20 votes.
Yeah, far and away the winner.
Thank you. Milady.
It's like anybody else stood a chance after that.
Most insightful design lesson shared, milk. And finally, most creative name, pampered cows. Okay. And so we have one thing left, and that is we'd like to have you assess how this went. This is the first time that we've done this. We've done this somewhat on the fly, but it's been a lot of fun. And so your feedback is really essential to us.